this video for Bio 181, Chapter 7, Cellular Respiration. We're now looking at the final stage in aerobic cellular respiration. And this is going to involve the electron transport chain and chemiosmosis and oxidative phosphorylation. These are all big words, big phrases that we'll go through. So electron transport chain. In this chain, it's basically a sequence of electron carriers that take electrons from NADH and FADH2, which have been capturing electrons from glucose in all the previous stages. And they give their electrons to proteins in the inner mitochondrial membrane. And those proteins, these proteins here, those are what we refer to as the electron transport chain. So they're going to take the electrons and they're going to use those electrons to power the transport of hydrogen ions across the membrane. That process is called chemiosmosis, or transporting ions across the membrane, and we're going to put them in the intermembrane space of the mitochondria and create a proton gradient. I know that's a huge mouthful. So we have, let's see, what's this next? Okay, so the electron transport chain. So I'm going to draw the mitochondria. So this is the organelle. That's the site of aerobic cellular respiration. That's the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. All right. In the inner membrane of the mitochondria, there is a series of proteins that are embedded in this membrane, this phospholipid bilayer. And this sequence here of proteins is called the electron transport chain. Okay. They are going to carry electrons. They're going to be given electrons here. Okay. They're going to be dumped off and they're going to carry them from one to the next, to the next, to the next. Okay. And they're ultimately going to give that electron to oxygen and make oxygen gas. The electrons are coming from the electron carriers NADH and FADH2. So these were made in the previous steps of cellular respiration. All of the energy from those carbon molecules, carbon-based molecules, was pulled off and brought here, okay, at least a lot of it. And why are we doing this, right? So we've taken all these electrons, we have them, now we're putting them on these electron transporters. Why? So the purpose is, I'm going to draw it again over here just because it gets kind of messy. So I'm drawing it again over here. Here are my proteins, okay? And as the electrons go through, these carriers are going to use that energy to pump hydrogen ions into this space, okay? So as the electron goes through, that energy is going to be taken off. The electron's going to lose a little energy, and then that energy is going to be used to create basically a hydrogen ion battery. We're putting a whole bunch of positive charges together, right? Remember, a hydrogen ion is a proton, right? It's a proton. So I'm putting together, or your mitochondria right now in your body, while you're watching this, all your little cells are doing this, okay? They're putting all these positive charges together, and they don't want to be together. They repel. So you're making a battery. They want to get away from each other. You're creating stored energy. That's what the electron transport chain is doing. Okay. So it takes NADH and FADH2, the electrons from them, Oh, so I said it backwards in the previous one. Shoot. Okay, I'm gonna have to back up. I'm not gonna redo this whole thing right now, but please erase, erase, erase. So here, so the final electron acceptor is oxygen. It is going to give it to oxygen, but I just want to show you that that oxygen is ultimately going to take them and be converted along with hydrogen ions to water. So it's going to be the O2 is going to combine with some hydrogen ions that are still floating around here. Okay, with some electrons that are coming off of the chain. The negative electrons join with the positive hydrogen ions. They join with water. Here we go. We're going to make some water molecules. Okay, hydrogen ions join with the electrons, join with the oxygen, making the water. Okay. So the oxygen is the final electron carrier. It's going to combine again to make water, FAD and NAD, plus will also be the byproducts of the electron carriers. Once they give off their electrons, these will go back to get more electrons and bring them back. So this process keeps going. Okay. So electrons provide high energy electrons to the proteins that are embedded in the inner membrane. The energy from these electrons is used to pump hydrogen ions into the space between the inner mitochondrial membrane and the outer mitochondrial membrane. So, So between the inner mitochondrial membrane and the outer mitochondrial membrane. At the end of the chain, the electrons are accepted by oxygen gas, and then water is produced as a byproduct. Okay, the final electron acceptor is the highly electronegative molecule called oxygen. Without oxygen gas to accept the electron transport chain stops and cells will switch to fermentation. So that oxygen at the end is essential, or we're going to switch over to fermentation, which is not going to give us as much energy. And we'll talk about that in the future. Okay. Oxidative phosphorylation, we have a proton gradient. So we have a mitochondria. We've got all of these hydrogen ions in this space here, a whole bunch of them. They want to get away from each other. Okay. That is going to power ATP synthesis. So we've got a molecule. It's going to be embedded in this membrane here. It's going to take those hydrogen ions. As it takes them through down the gradient, it's going to make ATP. Okay, so this is where most of the ATP is made. This molecule is called ATP synthase. Okay, ATP synthase, and that is really the powerhouse of the mitochondria. So chemiosmosis just refers to the movement of ions across the semi-permeable membrane. That's a vocabulary word. And this is showing that process. Here's the inner membrane. Okay. We've got the electron transport chain pumping the hydrogen ions into the inner membrane. As they're built up, they create a battery. They are then allowed to go through ATP synthase. And as they do, they make 28 ATP. So they're making a huge amount of ATP. So ATP synthase, that's the enzyme that catalyzes that reaction. It's a rotary motor. This is the overview. We're making 28 ATP. The enzymes in the inner mitochondrial membrane are the location. And here's where if we do the calculations, you have a little ATP that's built up along the way. but you have most of the ATP that's created through chemiosmosis. Okay, so let's just see. I was trying to rush through this one, sorry. So this one really just looking at the theoretical yield from each of the NADH molecules and FADH2. So the idea is all of that energy ends up making ATP, most of it through the process of chemiosmosis, some of it through substrate level phosphorylation. All right, and that is it. Whew.